give few housekeeping notes. First thing is kindly mute your mic throughout the webinar session to avoid interruption during the session. Questions can be posted in the chat box of this webinar platform. And finally is participants are required to fill up two different links for this webinar. Link 1 is for the e-certificate and the link 2 is for the webinar feedback. The links will be available 30 minutes after the session started. So without further ado, I would like to welcome everyone to the PPSM Webinar Series organized by Persatuan Pegawai Sains Malaysia. I'm Vidubala Balasegar, will be the moderator for today's PPSM Webinar Series entitled MPOX, the Laboratory Perspective. To begin our session, I'd like to briefly introduce our guest speaker from the Virology Section, National Public Health Laboratory, Malaysia, Sungai Buloh, Madam Ushananthiny Ravi. Madam Ushananthiny Ravi graduated from University Kebangsaan Malaysia with a bachelor's degree in microbiology in year 2005 and a master's degree in food microbiology on 2007. She also holds a postgraduate diploma in medical microbiology provided by SEOMED Traumed in 2013. Madam Usha joined MOH Malaysia on year 2000, serving as scientific officer in Department of Pathology of Hospital Tengku Ampuan Afzan, and soon after to Hospital Sultan Haji Ahmad Shah in state of Pahang, predominantly in the field of bacteriology and serology. Starting 2013, her career shifted to virology works in the National Public Health Laboratory Malaysia, focusing mainly on virus isolation and electron microscope sciences. Currently, she is the technical officer for General Virus Module in Virology section, covering several important portfolios such as MPOX, chikungunya, AGE, Ebola, rabies, and many more. Apart from that, she is a certified radiation protection supervisor, being actively involved in ISO 15189 as a document controller for 6 consecutive years now. And she is also a member in country's guideline preparation for chikungunya, Ebola, food water-borne diseases, and recently MPOX. She has also facilitated international projects and training by ASEAN and RPHL network, especially during the COVID-19 pandemics. It is indeed a great honor that she is able to deliver a talk for today's session. Thank you very much, Madam, for your acceptance for this PPSM's invitation. Now, I would like to invite Madam Ushananthiny Ravi to deliver her talk. Over to you, Madam. Thank you.
Thank you very much, Puan Vidu, for the very kind introduction. So I just want to confirm, can everyone hear me clearly?
Yeah, we can hear you, Madam.
Okay, so then allow me to share my slides. Yeah.
Madam, you can share your slides.
Yes,
No problem.
sharing. Okay, so salam sejahtera and a very good morning to all our attendees today to the webinar. So I am Ushananthiny Ravi, a science officer from NPHL Malaysia. So today I'll be talking about the very hot topic, MPOX, from the lab perspective. Okay, so this would be the overview of my talk today. It seems like a lot to be covered, but I will keep it as concise as possible. Okay, before getting very much into the lab perspective, Okay, I would like to share some general knowledge about the nomenclature of the monkeypox virus and also the disease itself. Okay, as we know, monkeypox virus was isolated in 1958 from the monkeys, captive monkeys in Denmark. So hence the disease name monkeypox. But to date, nobody knows where's the real origin of the virus. And now to avoid stigmatizing statements, towards the animals or the place of origin of the virus, a collective decision of experts through WHO has been made to change the name of the disease from monkeypox to MPOX. However, the virus is still known as monkeypox virus, but the variants has been renamed. So what we used to know as Congo Basin and also West African variants is now known as clade 1 and clade 2 respectively. Okay. Now I'll go into a brief information on the global MPOX case distribution by clade. Yeah? Okay, WHO has actually declared that MPOX is a public health emergency of international concern on the 14th August 2024. So this is due to the alarming number of cases detected outside of Asian regions and also the detection of a novel clade 1B. As of 15 September 2024, as we can see, monkeypox virus clades detected globally as shown in this map. Okay, outside of Africa, all the countries, most of the countries are affected by clade 2B, and in Africa itself, the variance is of the variation, sorry, is of clade 1B, clade 1 and clade 2. However, in this year, 2024, Sweden and Thailand has reported their first cases of clade 1B. So for Malaysia, we have detected a total of 9 positive cases in 2023. And as of yesterday in Malaysia, we only have 1 case positive for 2024. And through genotyping, we have detected clade 2B as the circulating variant in Malaysia. Okay, a quick glance on the MPOX disease profile. Okay, we all know that MPOX disease is a zoonotic disease, and the virus can be found not only in the various species of monkeys, but also in rodents, squirrels, pouch rats, dormice, and other animals as well. So the mode of transmission can be summarized into closed and prolonged physical contact with someone who is symptomatic, okay, through body fluid and also respiratory droplets transfer. Second one, contact with contaminated materials or surfaces, and the third one will be infected animals. Okay, as you can see, this disease can be divided into a few stages. Okay, starting from the incubation stage. The incubation stage is around 5 to 21 days after exposure, with an average of 6 to 13 days. So moving on to the next stage, which is the prodromal stage, which will last around 1 to 3 days. Okay, patients during this time will develop fever, headache, backache, myalgia, lethargy, and significant swollen lymph nodes or lymphadenopathy. So the next stage is the rash stage. Okay, together combined, stage 3 and 4 will last around 2 to 4 weeks. Okay, the rash will appear and it will slowly develop from macules to papules to vesicles, pustules, and also the last stage, scabbing or the crust stage. So as for the treatment, the disease is self-limiting, okay, and we need to support the patient to manage their symptoms and also to prevent complications. There are vaccinia virus-based vaccines available in the market, and some studies says that therapeutics or antivirals developed for smallpox may have some benefits against monkeypox because of their genetic resemblance, but there is no sufficient data to support this so far. Okay, let's get to know a little bit about the monkeypox virus itself. So the virus is belongs to Poxviridae family from Orthopox virus genus. Okay, it can be classified further into clade 1 and clade 2 with subclade 1A, 1B, and subclade 2A and 2B respectively. So what are the differences between clade 1 and clade 2? So clade 1 tend to cause severe illness with a higher mortality rate of up to 10% and a very increased transmissibility. So whereas clade 2 is a total opposite, it only causes milder infection with a mortality rate of less than 1% and also reduced transmissibility. So as for the genome, it is a linear double-stranded DNA with a size of around 200 kilobases, which encodes for around 181 proteins. And monkeypox virus is relatively a huge virus with a particle size of around 200 to 300 nanometers. So as we can see on our right, the electron micrograph, courtesy of Robert Koch Institute from Germany, the particle is said to be brick-shaped or mulberry-shaped. Okay, so now moving on into the lab component, we will start off with the biosafety and biosecurity measures. So monkeypox virus falls under the risk group 3 organism. So ideally, MPOX samples must be processed in the biosafety level 3 containment. Okay, just like what we practice in NPHL Malaysia. But we truly understand that not all lab has got these BSL3 facilities. So alternatively, we may process the samples in a biosafety level 2 containment with added procedures, with heightened safety and practices, just to make sure all the biosafety biosafety aspects are strictly adhered. Okay, and then the recommended PPEs will be disposable coveralls, double gloves, powered air purifying respirator or N95 mask, goggles or face shields, just as shown in the pictures. And the picture below shows one of our staff all geared up with full PPE to start work in the PSL3. Okay, as for the biosecurity part, the important objective is to
Sorry, madam, your voice is like not clear. I think breaking
give
and like going off and on. So
okay,
the
okay.
participants are complaining.
I'm trying to fix the problem. Can you give me just a few minutes?
Yeah, sure, sure. Thank you.
Hello, can you hear me now? Hello?
Hello, better, Doctor.
Yeah, better, better.
Better now, yeah? All right. Okay, so where was I just now? Sorry. Okay, so the personnel working with the virus must receive a very comprehensive training in biosafety protocols. This is very important because we do not want any cases of laboratory transmissions or any lab acquired infections. Next will be the biosecurity measures. So our main objective is to prevent intentional release and misuse of the virus. So we have to have control measures for that. Okay, next will be the recommended specimen types. Okay, the decision of what specimen to collect is basically relies on the clinical presentation of the patient. If the patient has vesicles or pustules, then lesion fluid or lesion fluid swab will be the best specimen. And to ensure sufficient DNA material is collected, swab 2 separate lesions using 2 different swabs, and you have to place it in one swab or in one container containing VTM or just the sterile container. If let's say the patient is in a scab or crusting stage, we can collect the scab and crust from 2 specimens taken from different locations also and put into the same sterile container. And if the patient is a contact and they are in a very early stage of symptoms and without rashes, we can go for tonsillar swab or nasopharyngeal swab. Just like we can collect the specimens just like we collect for COVID-19 testing. And please, just a little note, serum or blood samples are not recommended due to a very low viral load. Okay, this is because viremia usually happens at the early course of infection and it is of a very short period, and this may give us false negative results. Okay, let's talk about the most important part, which is the testing method for diagnostic purposes. So this is, we are discussing about diagnostic purposes. Yeah? So WHO highly recommends nucleic acid amplification testing, or NAAT, for detection of monkeypox virus. So PCR remains a gold standard for monkeypox virus detection and also in some cases clade determination. So why? Because PCR is highly sensitive, highly specific, and it is time saving and also easy to perform in our labs. Okay, to further determine clade or also to further identify the subclades, we can go for sequencing. And sequencing also gives us information about genetic mutations and also emergence of new variants. Okay, so the other PCR methods, PCR-based POCT methods that is used in different parts of the world, example, Gene Expert, but they always come with limitations, okay, such as manufacturing capacity, very low manufacturing capacity, and also delayed supply chain due to prioritization to the high-risk countries and also very high costs, up to 20 USD at times per test. Okay, so now we know what test to use, and we should also know that what is not recommended as a diagnostic tool. Okay, the first one would be virus isolation. We need appropriate experience, very high containment facilities, and also it is time consuming, and that is why it's not suitable as a diagnostic tool. Okay, moving on, electron microscopy. This is a very useful tool. It's a catch-all method, but unfortunately, we cannot distinguish between orthopox virus species, and we need highly skilled, very highly skilled personnel and also special EM setup. Okay, serology, there is always a risk for cross-reactivity with antibodies to other pox viruses or vaccine. So it may give us false positive results. And lastly, antigen-based rapid test kits. Okay, so far there is no sufficient convincing data on the kit performance. So, however, please take note that these are all very useful, very supportive tool for research purposes in conjunction or in correlation with the PCR findings. And in Malaysia, just a little note, for any testing kit used, it must be MDA certified. Okay, when it comes to lab diagnosis, WHO has clearly stated negative cases and positive cases, I know, for better understanding. So for orthopox virus, okay, we usually screen for orthopox virus and or monkeypox virus. So when the result comes out as negative, that means that suggests that monkeypox virus is not detected. However, If let's say there is further investigation is highly indicated, we can actually repeat the test or we can actually go for sequencing as well. Okay, for positive cases, the flow chart seems to be a bit complicated, but I can just summarize that if orthopox virus and or monkeypox virus PCR is positive, then it is confirmed as a MPOX case. So what we can do, like in reference laboratories, we can proceed for clade determination and subclade determination, either by PCR or sequencing. Okay, this slide shows the testing capacity, the current testing capacity in Malaysia, whereby we have around 8 government-based laboratories and also 6 private laboratories able to perform the real-time PCR for monkeypox virus detection, whereby MKAK and IMR can also proceed for genotyping. So the other laboratories are needed to send us the positive samples for genotyping purposes. Okay, laboratory turnaround time for monkeypox virus real-time PCR is 48 hours, and for genotyping is 5 to 14 days, as it involves more complicated steps and also very intense data analysis. Okay, this is just to share some, these are our molecular activities, for detection of monkeypox virus in NPHL Malaysia. So our routine diagnosis involves the real-time PCR for monkeypox detection. And if there's any positive cases, we will proceed for genotyping and, clade determination and subclade determination. And also shown here are the web-based tools and also applications which we use for data analysis and references. Okay, as for virus isolation for monkeypox virus, these are the beautiful cytopathic effect or CPE development of monkeypox virus in the Viro E6 cell line. Yeah, okay. So as we can see, the cells will, if I may show, the cells will be rounded and it will be detached from the surface of the culture flask as early as 24 hours. And just to emphasize here, all our culture works are strictly done in BSL-3 containment facility. Okay, next, this is just to share a micrograph of the MPOX virus detected by us in NPHL Malaysia. So this is by negative contrast staining and screening by transmission electron microscope. So what we have here is a brick-shaped viral particle with an average size of 220 to 270 nanometers. This is at a magnification of 52,000 times. Okay, and for more information on MPOX testing, please refer to our website. Okay, these are the part of my references for monkeypox works in NPHL Malaysia. And finally, I would like to extend my sincere gratitude to our MPOX team in NPHL Malaysia. Without the support of the whole team, we would not be here today. So thank you very much, and thank you everyone for listening. Back to you, Puan Vidu.
Okay, thank you so much, Madam. Thank you for the concise the monkeypox from the laboratory perspective for us today. So I would like to welcome questions from the participants. You are welcome to key in the chat box, or you can unmute your mic to ask the question directly to Madam Usha. So just to kickstart for the Q&A session, I would just like to check with Madam Usha. What are the key challenges faced by our laboratories in detecting the monkeypox outbreaks or monkeypox virus as well?
Okay, as for the key challenges, I would say because we are dealing with viruses and viruses mutate, so there's always emergence of new variants. So the key challenges is we have to, as a lab personnel, we have to be updated in the testing methods, especially for PCR. So we have to be alert of the primers that we use are up to date and they are able to detect the current circulating variants. So we have to like always up to date. That are the key challenges, I think, for PCR lab.
Okay, madam. Yeah, because virus keep on changing and keep on mutating, right? So they might have new variants and new, like for monkeypox. I wonder why they call it clade, madam. It's another question to be asked actually.
This one, I think it's from the WHO. The claim. It can be, virus, they don't have a standard. Usually it could be variants, could be strains, and all that. So it so happened that monkeypox is, they use clade for monkeypox.
All right. Okay, we have one question from Pusat Pakar Perubatan Norma Sarawak. Are all the lab listed using the same method analyzer for testing? Quite interesting. So just now, Madam got show us the
The
list of
list.
labs. Yes. So are they using the same method analyzer for the testing?
I can say that they are using the same protocol, but for their analyzers, I'm not very sure.
Because they are doing the PCR, so I assume they are using the same primers, right?
Yeah,
Across the
primers
lab.
and protocols should be the same, but if you ask me about the analyzers, I'm not quite sure.
Hope that answers the question. Okay. Another question is from Aminatul. It is possible to share the recording. Okay, that's fine. We will do it in our website later on. So for the other question that I would like to ask is, how actually we differentiate monkeypox from other similar pox virus infections in the lab?
Okay, if we see the workflow that I showed just now, we will test for orthopox virus, which covers all the pox viruses, which can infect
Okay.
human. So that is how it has been, we use that to differentiate. And
Are
we
we
also run
using a
for monkey.
nested?
No,
Sorry,
normal
no, we
PCR.
are not using monkey, but the primers can actually detect all the orthopox viruses, lah.
Yes, correct.
All right. Okay. Okay. Another question.
The
As
the
for
sound
sample,
is breaking a bit.
sorry. Can you hear me
Okay.
now? Okay.
Yes,
So,
yes.
okay, for another question from Anis Lee, she's asking, I think it's a she. As for sampling, any key personnel that we can refer? Okay, they want to know if we can refer to anyone that actually can have advice on the sampling method.
Okay, for sampling methods, okay, we in our website we will have the videos that covers all the for the PPE, for the sampling methods, for lesion and everything, so we can refer there in our
Alright,
website.
so,
Yes, there's a video.
okay, another
We are
question regarding
we are in mid of updating the video, so it will be available as soon as possible. We have an older version,
Okay.
but we are also
All right.
updating it. Yeah.
Okay. So there's another question regarding the sample as well. They said, can nasal swab with blood stain used for testing monkeypox?
Oh,
Nasal
so
swab.
detailed. Can
Nasal swab. I think nasal
blood
swab
stain.
is not a blood stain, and
Yeah.
it's a nasal swab. Is it suitable, madam?
Because we encourage to take nasopharyngeal swab, do they mean that along the way when they are collecting the sample, there's some bleeding? Okay.
I suppose that should be the question means.
Yeah, but
Let's.
yeah, if possible, avoid that. If possible,
Yeah.
avoid that. We just need a clear nasopharyngeal swab or tonsillar swab.
Okay, there's another question on sampling again. What is the best sample for forensic cases? if that case is coming in with suspected of monkeypox, so what is the best sample to be taken?
Okay, why do we suspect monkeypox in the first place? Because the patient has symptoms, right? Symptoms of rashes and all. So we probably could take the same kind of specimen from forensic cases.
Okay, but there is, if I'm not mistaken, I came across in the literature is that it can manifest as the pox, as the pus, or also it cannot have the pus, but they can still have the monkeypox. Is it true, madam?
Come again, Ponvido, with your question. What is it?
No, because the symptoms can be either manifested as the pustules, the pus, or it can be not manifested at all as the pus.
Okay, understood.
Yeah.
Understood. Because if let's say they didn't go to the rash stage, meaning they are in a very early infection stage. That means that is why we encourage them to get the swabs of the tonsillar and also nasopharyngeal, especially for contact cases.
Right. All right. So it will, in the end, it will still manifest as the pox coming out on the skin lesions, right?
Yes, later on, yes.
Yes. Okay. So there is another question again. For the personnel that can take the sample, do they need to have any certification, or medical personnel can take the sample? I think this should be anyone, right, who is a certified medical personnel.
Yes, that is, and with proper training and PPE. It's usually IKs, doctors.
Yeah.
Mm.
Yes.
Mhm.
So it is something similar like COVID situation, but it's slightly different because it's having the skin lesion, I suppose, right? All right.
Correct.
So another question from Miss Nami. For lab with minimal facilities, for example, with no BSL or damaged BSL, how do we handle this sample? Is there any minimum recommendation for us? Okay.
Damaged BSL.
Yeah, maybe they don't have the proper BSLs, level 2 or level 3, class 2 or class 3, sorry. So those labs that do not have this kind of facilities, what is your recommendation, madam?
Okay, I would recommend them to send to any nearest facility which can proceed with this test, because I don't encourage, if you don't have any, at least BSc class 2, please do not proceed. Please send to the nearest lab who can do that for you.
So they just pack it off and send it to the nearest lab that have the
Yeah,
facility.
that is my personal suggestion.
Okay, all right. Okay, another question. If I'm not mistaken, the vesicle or pustules is not easy to get the lesion, so the sampling might be challenging. Correct me if I'm wrong. I think this is the early stage of the thing manifesting on the skin. I think.
Yeah,
So is it?
so far we have been getting lesion swabs and no complaints about it. You just try to lah get, because that is the best specimen to be tested, because the viral load is very high there.
Okay. All right. Another question from Dr. Nadia. In a private facility, if we have the test kits, are we required to outsource the test for confirmation? I think this should be a yes. Right, madam?
They have kits. Okay, they have testing
The,
kits. Private facility, meaning are they private labs?
maybe it's a private hospital lab. Probably. I'm not sure the question from the
Okay.
person. Maybe the person can rephrase the question if they want the clearer picture.
Okay, okay, I got the answer. It's a private hospital lab. Okay, we have some criterias for the private laboratories to be accepted to perform the monkeypox detection. So you have to go through KKM to be a certified lab to proceed with monkeypox detection.
Yes. Okay. So another question, I think many more questions coming in. We still have some time, so no problem. So how much should we take for lesion skin sample? Is there any some measurement, for example, diameter of the lesion, weight, or anything?
There's no so detailed dimensions. It's just that you have to swab it vigorously, okay, just to ensure that a sufficient viral DNA amount is there in the swab. So you can use two different swabs to swab in two different locations of the lesion, and you can just put it in one single tube or container, sterile container, and send to us.
Okay, I think this question was answered earlier. The person is asking, will there be an updated video on taking samples? I think you just mentioned, right? You
Yes,
will have
yes,
a
yes. There will be. Mhm.
Okay. All right. Another question. Mode of transmission is by touching. Kenapa sampel untuk kontak perlu tonsillar swab or NPS macam COVID? I think this is for the earlier infection, right,
Yes,
madam?
you know why? Because the virus from mode of touching itu dia boleh bergerak to all these sorry,
Parts of
to
the
all these
body.
parts of the body. Okay, so that is where all the viruses are found. So mode of transmission is one part, and where the virus can be found is another part.
Okay. Another question here. If there is two lesion, genital and mouth, so it should be taken swabs from both sides with two swabs each, total of 4. So it should be combined in one or separate VTM according to site.
Okay, you can take swabs from different locations. It could be from your mouth or from the genitals, but you can combine. So in one swab tube, you will have two combined swabs. Is
Yeah.
that clear?
So I think I
It can be
suppose there
from
should
any
be.
location, any location.
Yes.
Mm,
So they can combine the mouth and also the genital one in one
yes,
tube, lah.
yes, can.
Okay.
Mhm,
All right. Wow. Okay. Since
mhm.
they are very, my participants are so enthusiastic to ask
Yes,
questions, I
I
think.
can see all
Okay.
the questions coming in.
Yeah. Okay. Is it safe to say then that monkeypox situation is controlled in Malaysia? I think definitely a yes, because it's not spreading like COVID-19, I
Yeah,
suppose.
it's not like a COVID. Yes.
Yes. Okay. Another question. If we want to send the sample to MKK, but due to transportation and logistic problem, there's some delay in the delivery of the sample. How should we store our sample? It is enough in 2 to 8 or need to put in -20?
Okay, this is a good question. Okay, within 1 hour of collection of samples, please refrigerate the sample. That means maintain cold chain 2 to 8 degrees. So this can be up to 48 hours. So after 48 hours, you can just freeze it in -20 degrees Celsius. So if the storage is going to take longer than that, please store it in -70 degrees Celsius.
So the storage would be it remains in the VTM, or are we going to put it in some other solution to store it in the -20?
No, it will be in the VTM. So the whole
VTM.
pack, yeah, the whole tube must be stored in a refrigerated this one.
Okay. If there is any question, please unmute your mic, or otherwise, please mute your mic, participants. Okay, so coming to the storage and transportation, actually, how does the sample collection and storage impact the accuracy of MPOX diagnostic testing? Is it like
Okay,
similar like other viruses, or we have some exclusion for MPOX?
no, there's no exclusion because for all viruses, it is very crucial for us to maintain the cold chain, and that is where the specimen quality is maintained. So it is very important for PCR testing, especially. So it's similar to all viruses.
Yeah. Okay. So regarding the lesion sampling, there's another question. Procedure, should it be taken using dry swab or gel swab? Okay. Maybe
It is dry swab. It should be a Dacron swab or polyester swab. It's not gel swab. I'm not very sure what is gel swab do they mean like a steward media or something? If yes, then no, we're going to reject that. So please stick to Dacron swab or polyester swab.
Okay, all right.
Mhm.
And okay, another question: is the test cost effective to be run in-house? In-house means maybe it's in the local laboratory, probably. Like, that's what I'm supposed to
Yeah, I think a
understand.
PCR, yeah, is widely used now, and it is cost effective as compared to other test methods.
Yeah, because I think compared to COVID, MPOX,
Mhm.
what I can see is MPOX is quite sometime it's already there. So the primer is easily available. So PCR, if you have the facility, I think
Yeah,
it's
why not? Mhm.
why not? Yeah, that's what I think.
Mhm.
Because COVID, we had a struggle to get the primer in the initial part, if I'm not
Mhm.
mistaken. We
Actually,
can't identify.
COVID helped us a lot to set up, uh,
Yeah,
even those who
PCRs.
don't have PCR tests started performing PCR tests. So we are quite well established in that.
Yes. So we
Mhm.
are quite ready for that if is
Mm.
another thing coming in, lah. So
Yes.
another question. The positive result will be released within 48 hours and the
Mhm.
clade genotyping will come in later. Is it? I think yes.
It
Because
is, yes. We for our requesters, we will report up to monkeypox detected, virus detected or not detected. So as for the clade and subclades, it's only for the public health purposes. So it won't be
yes,
reported.
reported. This is actually for surveillance purposes to
Yes,
see whether
surveillance and
which
public
clade
health
is.
decision makings, and all that.
Yes. So actually, madam, coming to this clade thing, can we
Mhm.
just slightly discuss about what is the main difference between the clade 1 and clade 2, or the Congo basin and the African basin? Can we just talk a little bit on that?
Okay, sure. As I mentioned earlier in my slides, clade 1 is actually, it causes a more severe infection. Okay, and it has a very high mortality rate, which is up to 10%, and it has an increased transmissibility. So clade 2 is the opposite of it, lah. So it causes milder infection, and the mortality rate is less than 1%, and it has a very reduced transmissibility. So
So
that's the
so far
difference.
in Malaysia is the clade 2, right? It's circulating.
Yes. Okay, fingers crossed. It will be only
Keep
clade
our
2.
fingers crossed. Yes. Okay, all right. So, okay, another question. May I know which is better, crushed sample in VTM or sterile container? Wow. Okay.
Okay, we do accept both. But for scab, scab, it should be sterile container, no VTM.
Yes.
Mm-hmm.
Yeah, because it's already a scrap. So we
Mm.
ideally put it in a sterile container.
Yeah, sterile
Okay.
container.
All right. Can the pustules or liquid, hold on, let me see this. Can the pustules or liquid sample taken using sterile syringe and pump it out into the VTM or sterile container? Okay. Wow, they can syringe
Yeah,
out the
why
sample.
so complicated? It sounds so complicated.
Yeah, that's the thing.
Yeah, because
Uh,
that is like invasive. It sounds like invasive, so we prefer to just swap directly.
okay.
Mhm.
So, madam, you want to share anything else on the laboratory's perspectives?
I think I've covered pretty much all in my slides. What we
Yeah.
do? Yeah.
Yeah, okay. So there's another two more questions coming in, and I will say I hope this would be the last two. So one question is it advisable for clinical personnel to use sharps to cut the lesion if the swab is unable to break surface of the lesion? Well, to that extent. Maybe madam can tell me, is it?
Because the instruction from WHO is to swap it vigorously. So the usage of sharp tools is always not encouraged, especially with monkeypox cases. So swap it vigorously.
Because it may come out like it can cause
splashy,
aerosol,
yeah,
aerosol or
aerosol generation. Yeah.
yes, so it's not advised. Please take note. Do not use sharps. You have to swab it vigorously. So no sharps allowed. All right. In our facility, the molecular and microbiology sections are combined into one. To conduct monkeypox testing, should we prepare a separate working area distinct from the main laboratory? I think it should be a yes.
Yes, it is and yes, because we have to have clean PCR rooms.
Yes.
Mm-hmm.
So you will not to have combined lab lah. This is not advisable.
Mm-hmm.
So okay, still coming in questions. We are almost 11:45 here. Okay, so for private lab, if we have positive cases for monkeypox PCR, is that mandatory to proceed genotyping testing? For now, I think yes.
Yeah, for private labs also, you have to send to us in NPHL Malaysia for us to go about it lah, for genotyping.
Yeah. So because we will want to see what's the clade circulating in our country, right?
Country,
So
yeah, we need that data. Mm-hmm.
we need the data, and eventually, if since the infection rate is not as high as COVID, we are advised to do a genotyping. What I think. So with that question, I think that's all for today's session. Thank you for the participants who actively participated in our session today. It was indeed a good opportunity for us to get in touch with Madam Usha, who is actually working hands-on with the monkeypox virus. So thank you so much, Madam. And if our participants or other members who have questions can get in touch to the National Public Health Laboratory for more informations. And kindly check their website for updated videos and updated protocols. Right, Madam?
Yes, correct. It will be in our website soon, very soon.
All right. So participants, please fill in your e-certificate link and also the feedback link so that we can come in with more interesting topics in our webinar series. So with that, I would like to thank our speaker, Madam Usha, and also thank our team, webinar team, and also thank all the participants for participating in our today's webinar. Thank you again, and
Okay.
bye.
Thank you, Panvidu and everyone. Thanks a lot. Yeah.